Good evening, students. Welcome to my lecture series on aquaculture. In continuation with my previous talks, I would like to take you through another discipline in aquaculture today, and that is to do with pearl culture. Now, before I begin, let me share my screen with you. Now, this is a picture of artificially cultured pearls. Now, the synopsis of my lecture will be under the following heads: Introduction and history of pearl culture. What are the different types of mollusk species that are used to produce pearls? What are the stages in pearl culture? The different types of implantation methods? And of course, a couple of questions to test our knowledge. To introduce you to pearl culture, we can say that pearl culture or pearls have been known to mankind since the beginning of civilization. Pearls are highly prized as gems for their beauty. These structures are secreted by the mantle, which is the outermost skin that is present in the molluscs, such as the pearl oysters, and the production of pearls is in response to irritation caused by external or internal stimulus that is brought about by sand grains, molluscan eggs, parasites, debris or detritus, and other foreign particles. What is the history of pearl culture in India? Our country has had the highest demand for pearls for setting it in jewelry. Pearl jewelry produced in India is famous world over. The pearl oyster fisheries in India are located in two main areas, one in Tamil Nadu and the other in Gujarat. It is present in the Gulf of Mannar in Tamil Nadu and in the Gulf of Kutch in Gujarat. Now, what are the animal species that produce pearls? The natural pearl-producing marine mollusk is known as Pinctada furcata or the pearl oyster. In our country, we have harnessed the potential of three freshwater mollusks or mussels, namely Lemmelidens marginalis, Lemmelidens coreanus, and Pariasa corrugata, which are known to produce good quality pearls. This is a picture of the pearl oyster, Pinctada furcata. We can see that it has an irregular margin, but it is known to produce some of the best pearls. And we should also remember that Pinctada furcata is a marine mollusk. This is a picture of the freshwater Lemmelidens, which is known as Lemmelidens marginalis. Most of the species belong to the genus Lemmelidens, and there are a couple of species that are present which can be used for artificial pearl culture. This is a picture of Lemmelidens coreanus, also used to produce artificial pearls, and Pariasa corrugata is another alternative species that is used to produce artificial pearls. Now, what is pearl culture? In nature, a pearl is formed when a foreign particle, namely a piece of sand or insects or some debris, enters into the body of the mussel that it cannot reject, and therefore, to prevent itself from being irritated, it suppresses the stimulus caused by this unwanted object by depositing a shiny coating on that particle layer by layer. This simple phenomenon is exploited in pearl culture practices. Generally, a natural pearl is small in size and irregular in shape. A cultured pearl is also a natural pearl, but it is made by human intervention by surgical implantation of the irritant or the stimulant in the form of a mantle graft and the nucleus for pearl formation. Now, what are the stages in pearl culture? At first, the mussels are collected, and once they are collected, the pearl culture procedure falls into 6 different steps or stages. Number 1, collection of the mussels. Number 2, pre-operative conditioning. Number 3, surgery. Number 4, post-operative care. Number 5, pond culture. And number 6, harvesting of pearls. Now let us take up each of these stages one by one. The first one is collection of pearls. To collect the oysters or the mussels, deep sea divers or divers, they go into the ocean bed or the river beds seasonally, say from February to September, and collect mussels. We can see here that this is a picture of a deep sea diver looking for mollusks. Once these mollusks are caught, they are transported in the form of nets that are made up of jute or nylon, and they are moved into the area for further processing. The next step is pre-operative conditioning. Now, what is pre-operative conditioning? The collected mussels, they are kept in aged water for 2 to 3 days in a crowded condition. That is what is known as pre-operative conditioning. Now, what is the meaning of aged tap water? Tap water that is collected and stored for a couple of days to remove the chlorine fumes from it is known as aged tap water. They are kept in a crowded condition so as to weaken the adductor muscles that keep the two valves of the shell tightly close to one another. When these mollusks are kept in a crowded condition for 2 to 3 days, we find that this conditioning weakens the adductor muscles and the valves of the shells open slowly and helps in easy handling of the mollusk for surgery. So once the collected mussels are brought in for processing, we find that they are kept in this nets for 2 to 3 days to weaken their adductor muscles. The next stage would be implantation of the irritant, and that is known as mussel surgery. Now, depending upon the place of surgery, there are 3 types of implantation methods. Number 1, mantle cavity implantation. Number 2, mantle tissue implantation. And number 3, gonadal implantation. Now, the materials that are required for mussel surgery are the beads of the nuclei, which are usually made up from the molluscan shells or calcareous materials. Let us look at each implantation method one by one. The first one is known as mantle cavity implantation. Now, the mantle is an external fold of skin that is secreted by the muscles. The space between the body of the mussel and the mantle is known as the mantle cavity, and here is where the implantation will take place. Now, in mantle cavity implantation method, beads that are of the size 4 to 6 in diameter are inserted into the mantle cavity region of the mussel after opening the valves of the animal and separating the mantles of the anterior sides from the shell. Implantation is made in the mantle cavities of both the valves. After placing the beads in the desired place, the gaps created during implantation are closed just by pushing the mantle onto the shell. Here is an image of the surgery in progress. We find that an apparatus is made to hold the animal whose shells or the two valves of the shells have opened up because of preoperative conditioning. And with the use of these forceps and needles, we find that a bead can be inserted into the mantle cavity. This method is known as mantle cavity implantation. The second method is mantle tissue implantation. In this method, the collected mollusks are first divided into 2 groups. The first group is known as the donor muscles. The second is known as the recipient muscle. The donor muscle donates the graft that is going to be implanted, and the recipients are those muscles which will receive the grafts from the donor muscle. In this implantation method, the first step is the preparation of the graft, which is composed of small pieces of mantle tissue. So, the mantle is removed from the donor muscles and they are cut into small pieces. This is done by preparing a mantle ribbon by taking a strip of the mantle tissue along the ventral side of the muscle from the donor muscle. So from the ventral part of the animal, a thin strip of the mantle is cut and made into small pieces. Once the graft is made from the mantle, the tissue implantation is of two types. Number one, non-nucleated implantation, and number two, nucleated implantation. In non-nucleated implantation, only the pieces of the graft from the donor muscle is introduced into the pockets that are created in the ventral region of the muscle. In the nucleated method, however, the graft piece is followed by a small nucleus, which could be a bead that is introduced into the pocket. The third method of implantation is known as gonadal implantation. And as the name suggests, we find that the implantation of the graft is made in the gonads or the reproductive organs. This procedure also involves the preparation of the grafts from the donor muscle. First, once the grafts are prepared, a cut is made at the edge of the gonad or the reproductive structure in the case of the muscle. Then, a graft is inserted into the gonad, followed by the nucleus, so that the nucleus and the grafts are in close contact. Care is taken in such a way that the nucleus touches the outer epithelial layer of the graft. Once the implantation of the grafts is completed in any of the three methods, the next stage of pearl culture is post-operative care. Those muscles that have undergone surgery or the implanted muscles are kept in a holding area that is known as the post-operative care unit in nylon bags for about 10 days. The 10 days period is to examine and observe whether the muscles have been able to withstand surgery. And during these 10 days, they are provided with antibiotic treatment for the healing of the wound and supply of natural food. The holding areas are examined daily for the removal of the dead muscles. Now, this is an image of the holding area or the post-operative care. We find that there are separate nylon bags in which the muscles that have undergone implantation are placed and provided with antibiotic treatment along with natural food. The post-operative care is for a period of about 18 months to 24 months, during which the pearl is synthesized by the animal. The next stage of pearl culture is the harvesting of the pearls. At the end of the culture period, the muscles are harvested. Now, individual pearls can be removed from the mantle tissue or gonad of the live muscles with no death of these muscles, and they can be utilized once again for pearl production. Whereas, if the implantation takes place by the mantle cavity method, then the muscles have to be sacrificed. This is an image that shows the harvesting of pearls in the pearl oyster. This is the marine pearl oyster, also known as Pinctada furcata. We can see that it has synthesized 2 pearls. This is another image that shows the end process of pearl culture, where the pearls are harvested. Now, depending upon the size, the luster, and the texture, these pearls are harvested, they are sorted, and they are sent to the market, or they are sent to the jewelers for being utilized in jewelry making, etc. So with that, we come to the end of a brief chapter on pearl culture. I request all the students to go through these questions. I will read them out one by one. What is natural pearl culture? What are the common species of molluscs used for artificial pearl culture? Name the marine and freshwater pearl producing mollusks. List the stages in pearl culture. Describe the process of muscle surgery in pearl culture. There are 3 processes, and I want you to pay particular attention to the nucleated and non-nucleated method of implantation. And of course, there is an essay type question where you have to summarize whatever you have learned about pearl culture by writing an essay on pearl culture. Now, I will attach this presentation in my Google Classroom. You all have the code for the Google Classroom, and the class is going to be conducted on the 6th of July. I ask all the students to attend that session. You can make use of this presentation. Assignments have been provided. Please answer them. If you have any doubts, please post your queries either on my WhatsApp or in the Google Classroom so that we can discuss for the benefit of all. Thank you.